Hello friends, today we will discuss about the palynology. Palynology is often insisted that it applies only to the pollen and spores, or more specifically even only to pollens and spores of the embryophyte or embryo producing plants. It is true that Hayden Williams had that in mind when they coined the term palynology, a word that is derived from the Greek word which means I sprinkle, which suggests of the fine meal, and it cognates with the Latin word pollen. Pollen means fine flour or dust. However, that is not the way most palynologists use the term. Instead, they use pragmatically based working definition, which says in effect that palynology consists of the study of the organic microfossils that are found in our maceration preparation of sedimentary rocks. That is, what my net catches is a fish. This means palynomorphs, the microfossils which are the subject matter of the study. It consists at least partly of very resistant organic molecules, usually sporopollenin, chitin or pseudochitin. There are some few exceptions to this also. Palynomorphs are also by common consent in approximate sizes of 5 to 500 microns. Many megaspores are larger, and some seed megaspores of the late Paleozoic are much larger. Species of the tuberculate sporitis are reported by some workers, and they can be even up to 3 millimeter. And cystosporitis megaspores, they can be even larger, they can be 3 by 8 millimeters. From a general and a pragmatic point of view, such huge spores are beyond the boundaries of the palynology. In megafossil paleobotany, that is not meant in any way to denigrate the study of the megaspores. On the other hand, nanofossils are not palynomorphs on the two scores. First, they are calcium carbonate consisting, and hence they are destroyed by the dilute hydrochloric acid or HCl. We usually employ as first treatment in the palynological maceration of sediments. Secondly, they are also too small, prevailingly less than 5 micron. Diatoms are not palynomorphs, although they are in the right size range because they are usually siliceous and destroyed by the hydrofluoric acid or HF. That is the major weapon in the palynological armory. The same is true of the phytoliths, usually consisting of silica or calcium oxalate produced typically by grasses and sedges. They are very useful in various forms of environmental construction. These are also in the correct size and dissolved by the acids in the conventional palynological maceration. A curious oddity is the report of the spores and pollens that are apparently permineralized and lack residual sporopollenin. According to our definition, such fossils through spores and pollen are not palynomorphs because they would not appear in the palynological residues prepared with hydrochloric acid, hydrofluoric acid, etc. Another problem with our definition is typified by some other works in which chips of the sedimentary rocks are very carefully and gradually treated with the hydrofluoric acid with no crushing of the rock fragments, no centrifugation, as little agitation as possible, no harsh chemicals other than HF, and handpicking of a very fragile specimens out of water with pipettes and other tools. Work with such specimens is certainly marvelous paleontological science and is very important, but their study is not strictly speaking part of the paleopalynology because fossils are not robust walled and therefore are not fish caught by our nets. The subject depends on obtaining whole assemblages of the abundant, at least relatively robust world specimens, data from which we use for stratigraphy, paleoecology and related matters. This is just a fact and does not denigrate the importance of fossils such as the Tapania specimen illustrated in conjunction with the palynological studies. Palyno debris is the organic matter found in the palynological preparations along with the palynomorphs. Even though not consisting to any significant extent of sporopollenin or chitin, some of it is more or less structured plant material such as charcoal, cellulosic tissue fragments that is escape the hydrolysis or acid digestion. Some of it is amorphous. Palyno debris is now an important subject matter of palynology for its relevance to the question of palynofacies environmental relationships. Charcoal in particular is a very abundant and frequent constituent of palyno debris and can be analyzed as to quantify and type, potentially revealing much about the environment at the time of deposition. In the nutshell, we must say that in the organic matter is basically a degraded part of the organic compounds, and usually in an organic matter, especially concerning plant parts, what do you like to expect? You can like to expect the spores, you can like to expect the pollen, you can like to expect the charcoal, you can like to expect the cuticle of the leaf tissues, but uh, whenever you are in a position to identify the individual components of that organic matter, this is called as structured organic matter. But when everything gets degraded, then you will not be able to identify a cuticle from a pollen, and then from the spores everything becomes amorphous. So, that is not a structured organic matter, that is the amorphous organic matter. So, palynofacies is broadly of two kinds, it is structured and amorphous. Now, in the next part of our discussion, we will be discussing about some stratigraphically significant palynomorphs. The first of which is Acritarchs. The Acritarch ranges from the Proterozoic to the present. The term means undecided or doubtful origin. It was introduced by Evatt in 1963 as part of a package to replace an older garbage term. Hystricospherids or spiny spheres, which also by analogy to the genus Hystricosphera, weasel, a synonym of a spiny spiritus. Many of the former hystricospherids were shown by Evatt and Wall to be dinoflagellate cysts. The hystricospherids which could not be shown to be dinoflagellate cysts were then left as acritarchs. However, the term now includes a very large range of the presumably algal bodies, mostly marine, but there are many brackish water or freshwater forms, including probable green algae echinids and the hypnozygotes, ranging from less than 10 microns to more than 1000 microns, but mostly they range in size between 15 to 80 microns, well within the palynomorph size range. The wall contains a sporopollenin or a very similar compound. They may be more or less ciliate, scabrate, spiny, reticulate and nearly bewildering array of the other sculpturing type. They first appear in the Proterozoic as simple, more or less spherical and featureless monads about 30 microns in diameter. Some palynologists follow Diver and Peat in separating off such bodies lacking spines, plates or any other features suggesting algal affinity as cryptarchs. However, it is yet more common to include the types of the one category acritarchs, Acritarchs, they range to the present time, but their greatest significance and high point of abundance was reached in the early Paleozoic. Tuppan shows that after the early Jurassic, spiny acritarchs are rare and unimportant, and though lysospheres and some other forms continue to present, acritarchs as a whole are not an important factors among extant phytoplanktons. The statement in Flug and Rees finally disappear in the Pleistocene is, however, too extreme. There are even some lysospheres and acanthomorphs in the modern sediments also. That means presently we are dealing with that part of the palynology which is not strictly spores and pollens, and they are together called as palynomorphs. Palynomorphs include all such forms whose biological affinity is known and also those forms whose biological affinity is not known. Next in the sequence comes as Chitinozoans. Chitinozoans range in age from late Cambrian to latest Devonian. These are pseudo-chitinous palynomorphs for which there have been several suggestions as the source organisms. One of these is graptolites, for which the relationship of the arguments are circumstantial but interesting, that is chemical similarity, frequent association, close agreement and stratigraphic limits. The suggestion of fungal relationship given by some like Loken in 1981 seems improbable. The best known experts on the group at the present is given by Paris et al. 2004 will go only so far as the eggs of the soft-bodied marine metazoans. They first appear in Cambrian rocks, are most abundant in the Ordovician and became extinct by the end of the Devonian time. They are found only in the marine rocks, that means they are completely marine. Their presence shows that the rock is entirely marine unless revoked. They sometimes occur in chains, but usually it also occur as single individuals also. Because of their thick, more or less opaque walls, they are usually best studied by the scanning electron microscopy or SEM. Although the light microscopy study of the broken surfaces frequently reveals important structural details. Because they are not represented in the abundance often seen for the spores and pollens and acritarchs, about 102 per gram range instead of the 103 to 105, that hundreds of specimens per gram of the sample of the larger samples must be processed. For this reason, because large specimens are easily broken, somewhat different processing techniques must be employed to study the chitinozoans. Next is the scolecodonts. Scolecodonts is another kind of the palynomorphs which is not a typical spore and pollen, and these scolecodonts, they range in age from lower Ordovician to the present. These are chitinous mouth parts of the Polychaetus, mostly marine annelid worms. Although they range from Cambrian to the present, they have been mostly studied in Paleozoic rocks. As is truly true for the chitinozoans, scolecodonts mostly occur in the range up to 102, more than 100 per gram, and require different processing techniques from those of the spores and pollen. Though fragments are frequently encountered in the slides from the conventional macerations. As pointed out by some workers, the mouth lining parts from one worm may exist united or dispersed, and the dispersed parts may not at all be alike. The taxonomy in this case therefore is difficult. Next is the microscopic algae and algal parts. These microscopic algae and their parts, they range from Ordovician to the present. Botryococcus is a very common occurrence, it is a very common example, and it is a colonial alga occurring in a wide range of the freshwater to brackish water aquatic environments. The walls of the colonies that are preserved, apparently it consists partly of the hydrocarbons, the hydrocarbon mineral corugnite. It consists largely of the Botryococcus colonies. There is some evidence that the walls also contain sporopollenin, although another species now regard Botryococcus colonies as representing Botryococcus browni cuisine, which would be, if this is correct, a candidate for the oldest surviving species of the Plantae, ranging in age from Ordovician to the present. Nicholas has shown that Botryococcus, both extant means the present day, and the fossil, possesses an extraordinarily diverse suite of organic compounds, stratigraphically and paleoecologically almost worthless. Botryococcus must nevertheless be treated here as it is found so commonly in the palynological preparations. The other principal colonies or colonial alga occurring as palynomorphs is Periastrum species, the various species of which range from early Cretaceous to the present. Although it is multicellular, it is more precise and technically correct to call it as green algae synobium, as the number of cells is fixed at the origin of the organism. The resistance to the biodegradation of Periastrum is one of the puzzles of the palynology. The walls seem so finely delicate and cellulosic and should be hydrolyzed quickly. Obviously, the walls must be impregnated with something additional, presumably the sporopollenin. The various species of Periastrum are all freshwater forms. Sporopollenin containing microscopic parts of the various green algal forms such as Zygnematacea, zygospores and Parasinophyceae, Phycomata are found occasionally as constituents of the terrestrial and nearshore marine sediments of Paleozoic to the present age. For an introduction to the various other microscopic algal bodies that appear from time to time as palynomorphs, parasynophyte algal Phycomata such as the Tasmanitids are also commonly found algal palynomorphs. Then in the series comes dinoflagellate as the next very major kind of the palynomorph. The dinoflagellate range from Cambrian, Cambrian is little doubted, to the late Triassic to the present. Usually they are found to evolve after the Permian Triassic mass extinction, but the similar morphologies are found in Paleozoic rocks as well. Sporopolleninous cysts of the dinoflagellate are common from the late Triassic rocks to the present, mostly in the marine sediments, but also in sediments deposited in the fresh and brackish water. The range problem indicated above has to do with the difficulty of proving that a given cyst is a dinoflagellate. This requires certification of the presence of the dinoflagellate type aperture or operculum called as arcupile and or dinoflagellate plates and related morphological features. So, dinoflagellates are basically plated organisms with the arcupile, which is a aperture, and then it is covered by some lid. So, all these features are to be identified before any palynomorph can be regarded as the dinoflagellate. Many Paleozoic cysts from as early as the Cambrian may be dinoflagellates also, but the required proofs are not present in sufficient convincing manner. Molecular and other chemical studies in recent years strongly support the pre-Triassic existence of this group. The work is mostly carried out by the Hackett et al. In 2004. On the other hand, many brackish water or freshwater dinoflagellate cysts are more or less featureless, bags that require examination of thousands of specimens to prove that a dinoflagellate made them. For example, in the Black Sea Deep Sea Drilling Project, that is DSDP course. We have discussed in this lecture many of those palynomorphs which are not true spores and pollens, but they comes under the realm of under the bracket of the palynology. I meant to say that all the palynomorphs are not only spores and pollens. We had many of the forms which have evolved in the geological past and they exist even now. Some of them have evolved in the geological past and they became extinct in the geological past for no time to come back, and these are also palynomorphs. So, palynology is basically the study of the spores and pollens plus the palynomorphs. Palynomorphs includes all those forms whose biological affinity is known and also whose biological affinity is not known. We will discuss about the spore pollen type of palynology in the next series, in the part 2 of this lecture. So, we will be meeting again in the part 2 of this lecture with the spore pollen or embryophyte type of the forms to be discussed in detail. Till then, thank you very much.